Hi everybody, welcome to another Chats with Children. I hope you're safe and well. Today I'm delighted to be joined by Samantha Barichievy from AstraZeneca and Luca Laraia from the Technical University of Denmark. And the reason they've joined me today is they are both track organisers and chairs for the upcoming ELRIG Advances in Cell-Based Screening event, which is going to be held at the AstraZeneca site in Gothenburg and runs from the 10th to 12th of May. And so we thought we'd have a quick preview of some of the topics that are going to be covered at that particular event. So Samantha, Luca, it's lovely to see you both. How are you?
Great. Very nice to be with you. Thanks for the invitation.
Thank you.
Lovely. Fantastic. Now, before we start talking about the event and some of the topics which are going to be covered, would you mind giving viewers a quick background on your own role? So Sam, would you like to start?
Sure, I'm happy to do that. So I am born and raised in South Africa, but moved to Sweden 8 years ago, and this is very much home. And I came over as a scientist specifically employed to make cell models for drug discovery. And that's what I do every day now. I lead a team who makes those cell models for early drug discovery.
Brilliant. And Luca?
Yeah, so I'm Italian, but I've been all over the place in Europe. I grew up in Austria, studied in the UK, worked in Germany, and then finally set up my own research group at the Technical University of Denmark near Copenhagen. And there I lead a research group in chemical biology. So I come from an organic synthesis background, but we use chemical tools to study biological systems.
Right, fantastic. So look, there you go, viewers. We've got a very diverse panel today. So my very first question, very simple, as a layperson, is what is cell-based screening and why is it important? So who would like to take that?
I'm happy to give that a bash.
Oh, go for it.
So cell-based screening in drug discovery, what we're doing is we're hunting for new drugs that we can use to treat your disease of interest. And a lot of the way we've done that historically is by looking at little molecules in a test tube, little pieces of protein, little pieces of RNA in a test tube. One thing we've not been able to do for technical reasons is expand that to actually looking in a cell and actually hunt for drugs in a cell or screen for potential new molecules in a cell. And that's essentially what cell-based screening is.
Right, okay. And Luca, would you like to add anything to that, or has Sam covered it?
Yeah, no, that's definitely it in a nutshell. But I think I guess one of the sort of things that's sort of happening right now, and maybe we can come to that later, is that we are now these days sort of leveraging the power of sort of microscopy, so being able to see sort of cells and sort of also subcellular sort of compartments and parts of cells very sort of clearly with sort of the more modern advances in microscopy. So that's also something that it will certainly be covered in the meeting, and yeah, it's pretty sort of big interest at the moment.
Well, that's a perfect segue, actually, Luca, that you've just given me. So you mentioned the meeting there, and actually, I know you're looking after specific tracks at the meeting. So, Luca, why don't you just talk about the track that you've been covering and putting the program together for?
Yes, so actually, the meeting runs over 3 days, and the third day is the one sort of I'm responsible or partly responsible for. And that is actually organized by the Nordic Chemical Biology Societies, which I'm also a member of. And so it's maybe slightly different to the previous 2 days because it has a slightly more sort of chemical biology rather than pure sort of cell biology angle. So within that context, we've invited three speakers to join us, and 2 of them are sort of experts in so-called chemical proteomics. What this means is that they use basically powerful advances in mass spectrometry, where basically you can individually follow the fate of in theory at least, every single protein in a cell at sort of different points in time or after different treatments with potential drug molecules. And this is really another powerful advance that has come through in the last sort of decade or so, which we definitely couldn't do before that. So we have Ben Cravat from Scripps in California coming, and he's giving the keynote talk. And we also have Laraia Piazza from Max Delbrück in Germany, who is also a sort of mass spectrometry expert. So they are doing really exciting work in this field. But we also have the third speaker, which is it's much closer to the theme, I guess, the general theme of the meeting, Ola Spjuth from Sweden, and he's talking about these kind of sort of advanced imaging techniques and analysis of microscopy images that I mentioned previously. So that's also, we're very excited about that.
Fantastic. And Sam, your track specifically, what does that cover?
Yeah, so I'm actually helping to co-organize a track, which is bookending the conference from the other end. So it's session 1 on day 1 of the conference. And our track is specifically titled Enabling Advanced Cell Models in Profiling. So one of the key things about drug discovery, like I said, and using cells for finding new drugs, is we can do a lot with cells that are grown flat in 2D. It's really, really difficult if we want to use cell models that are much more relevant, closer to a patient. And there's a lot of technical reasons for that. Actually being able to isolate them, being able to edit them so that we can turn them into a tool we can use, being able to scale them up so that we've got enough of them to actually use when we're finding new drugs. And these are the key things that we're going to be covering in the session on day 1. One of the really cool things that we're going to be focusing on is the diversity of genetic backgrounds that we're actually wanting to use in cell models. Historically, we've been really, really poor at that. We go back to the same cell types over and over. So one of the new things we're focusing on in this session is through our keynote speaker. So her name is Janine Schofield, and she'll be joining us from South Africa, actually. Her team specifically generates liver disease models. So these are little livers in a dish, but specifically taken from different Black African patients with very diverse genetic backgrounds. So we're hoping to sort of start that conversation in our track on day 1. We're going to be moving on to 3 additional speakers who are also going to be talking to us about how can we scale up these sorts of cells? How can we actually, as Luca touched on, how can we build imaging-based readouts that give us lots of information, multi-parametric information, we call it, or multi-dimensional information in a miniaturized format? And how can we then take all of that information and feed it back to understand the biology of the data? So we've built our track around trying to have a full circle conversation of how we can make advanced cells, use them, get data, and feed it back to the beginning again.
Well, no, that sounds really, really interesting. I mean, actually, I found it really interesting you talked about the fact that you have a speaker talking about sort of Black patients and the different gene types, because I know a lot of talk's been happening since COVID about clinical trials being very biased in one way of only having certain types of patients being used in those trials and not really reflecting the diversity in the general population and the global population as well, and how that impacts, in fact, the results that you get. So that's really, really interesting that you should bring that up as well. So putting aside that you are the organizers, but if you were an attendee coming to this event, what are some of the highlights apart from your own, obviously, talks that you've organized? So you're a bit biased, so you're going to say they're fantastic anyway, I know. But apart from those, what else are you looking forward to at the actual event? I mean, Luca, would you like to touch on that first?
Yeah, I think probably sort of the breadth of the science that's actually covered. I mean, I think there's several different sessions that cover very different topics. We've touched upon imaging. There's actually a whole other session in addition to the one that I'm chairing on kind of advances in sort of mass spectrometry to really study sort of individual protein levels across all possible proteins in a cell with drug treatments with in different disease states. So that's something that I'm certainly looking forward to outside of sort of my own session. But I think anyone who the topics are so broad that anyone who works in sort of drug discovery from any point of view should be excited about this thing.
Fantastic. And what about you, Sam?
I agree. I think just to build on what Luca said, there is another track also dedicated to oligonucleotides. So these are little RNA or DNA pieces as opposed to sort of classical bits of chemicals, which is a whole new area that we're sort of delving into in terms of trying to find new drugs. So that's very exciting. And I think from the softer side of things, we're really excited that we're physically going to be in touch with people again. This is the first face-to-face conference that we're going to be hosting in 2 years. So I think that's something really exciting to look forward to, that we'll be in this lovely space. Hopefully the sun shines and we can actually all be in the same rooms together again. So that's something I'm really looking forward to.
Yeah, well, I agree.
Well, I was going to actually ask you, give me a last picture why people should be there. But I think you both have summed it up very well in terms of your own little sessions just then talking about the event. So all that's let me say, so I wish you both lots of success and have a great time at the event in a couple of weeks. I won't be there myself, unfortunately, but I know ELRIG always put on a great event. And what I should say, viewers, is as well is that the event is free. So the link to register will be above this video. So if you'd like to know more, then please click on the link and they'll have the whole program on there and you can register, and it's completely free. And if you've got any questions for Luca or Sam based on what they said, because they're the experts, I'm certainly not, as you can tell, then please put them in the comments below the video and I'm sure they'll be more than happy to answer any questions you've got as well. So Luca, Sam, thank you very much for taking time out to talk to me today. It's been a pleasure meeting you both, and I genuinely wish you a fantastic time in a couple of weeks at the conference as well.
Thank you very much.
Thank you
very much.
And thank you, viewers, for watching. And until next time, as always, stay well and stay safe. Bye-bye.